Jeanne and Mara, and Jeanne is actually the leader in the fatigue and exercise group in Eastbourne, so the University of Brighton Sports Science Department. So for you who don't know, fatigue in multiple sclerosis is one of the most common symptoms in the disease. However, even though it is the most common and debilitating symptom as it affects people's day-to-day life, the root cause of fatigue is unknown. So there's many theories to do with different parts of the body, but there's no sort of holistic explanation for how fatigue arises in this disease. So previous research has shown that people with MS have higher levels of central fatigue, and this is in comparison to peripheral fatigue. So peripheral fatigue is anything that affects the muscles directly. However, central fatigue is anything to do with the nervous system. So we'll see anything north of the neuromuscular junction is central fatigue, and this is where MS have a higher level of fatigue, which makes sense because the disease affects the functioning of the nervous system. And perception of fatigue is also enhanced in people with MS, and this is mainly to do with the theory of interoception. So their predicted bodily states and expected bodily states is a mismatch in between what those are, and that arises in an increased sense of fatigue. And most research at the moment in fatigue and MS, it's very much based on sort of a single level. So you can study fatigue at multiple levels. You have perception, but then also fatigue can be studied at the cerebral level, the spinal level, and the muscle level. And existing research normally just focuses on one of these or two of these, and with or without perception of effort being measured. So our study is going to add to literature as we're the first ones that are going to measure fatigue at several levels. So we're going to be measuring fatigue from the brain, the spinal cord, and the muscle. And also with the addition of subjective markers of fatigue, so how they perceive the fatigue that they are experiencing at rest and during exercise. And comparison of all of this data will allow us to assess where the origin of motor fatigability is in people with MS. And we are also comparing it with people with healthy controls. So we'll have 30 people with MS and then 30 healthy controls. So we'll be able to assess the development and profile of fatigue in MS compared to what it should be. So our protocol is we're going to be investigating fatigue by inducing fatigue. So we're going to be inducing fatigue with the wrist extensor muscles. And the reason why we've chosen that is because we're restricted space-wise in the MRI scanner. So we're going to be fatiguing people in the scanner, and the wrist extensor is the biggest muscle group we can fatigue in the remits of the MRI scanner. So visit 1 will be fatigue with structural, functional, and MRS scans of the brain. And then visit 2 will be using techniques called transcranial magnetic stimulation, which assesses fatigue from the brain to the spinal cord to the muscle. And then peripheral nerve stimulation, which will measure fatigue from the motor unit to the muscle. So the fatiguing protocol that we've designed, we had a few limitations on how we could design this protocol because we're limited to closed loop exercise because of the scanning. So normally in fatigue studies, you'll see exercise until exhaustion. So just as long as you can until you fatigue, if the tasks fail. But as we're doing it in this scanner, we've got to have set blocks of time. So we've got to design an exercise that induces fatigue in a set amount of time. So we have decided to do a 3-minute maximal contraction. So that is as hard as they can for 3 minutes, sustained, no rest. So we'll see a drop in force sort of like around profile like that. Then we'll have 5 minutes rest as it's quite a demanding exercise. And then the sustained contraction after will be set at 10% below the critical torque. And critical torque is a physiological set point where if you work above that intensity, you'll see task failure occur. but if you're below it, then you can sustain that exercise for as long as possible. And that point also sort of determines what fatigue you see. So we will see more central fatigue closer to the critical torque. So that's why we've chosen just below the critical torque, so they should be able to sustain it with no problem, and we should see an enhancement of central fatigue, which is what we want to see in people with MS, because that's where we know there is differences. And to do this, we've designed 2 wrist extensor rigs. And the premise of these is the wrist will be secured in a set position, and then a force transducer, which will measure the force produced by the wrist extensors, will be adjusted into place. And we've got one for the MRI visit, so that's completely metal-free and safe to go in the MRI, and that will be positioned just across the chest. And then the second one is the TMS and PNS visit, so it's just a bit more robust because we're measuring extra force in that one. So visit 1 will be the MRI scanning, and we have chosen to do arterial spin labelling, and this works by marking blood in the carotid arteries, and then we take a scan of the brain to see where that marked blood has travelled to, and this gives us a direct sort of measure of blood perfusion and cerebral blood flow. And we've chosen this because it's been used in previous fatiguing studies. And also, if we wanted to use BOLD, the problem is you have to use intermittent exercise. You'd have to do, say, 3 seconds contracting, 2 seconds relaxing. And with people with MS, they struggle to produce torque, produce force sort of in that routine. So we're sort of designed exercise that's going to be easier for them to follow, and that's why we're doing arterial spin labelling. We're also going to be measuring glutamate in the brain, and specifically in the anterior cingulate cortex. And the reason we've chosen this area is it's involved in interoception, which is a process that is related to fatigue, and it also is involved in sort of how the perception of effort, the perception of fatigue arises. So it's that mismatch between the bodily states. And we'll be getting glutamate at rest, arterial spin labelling data at rest, and then we will go into task ASL, so that'll be the 100% contraction, and we'll get dynamic data from that exercise. Then immediately we'll get post ASL, which will be sort of a fatigue measure. Then we'll go into the sustained contraction, which enhances the fatigue and maintains the fatigue, and then we'll get post glutamate, so post fatigue glutamate levels, and then a recovery ASL. And that should give us plenty of data. And we'll also be collecting sort of how the participants are feeling in terms of fatigue, so ratings of fatigue and perception of effort. And visit 2 will be the transcranial magnetic stimulation and peripheral nerve stimulation portion of the study. So we will be collecting EMG data, so how the muscles are activated of the extensors, and also the flexor, but that is mainly just to make sure that's not activating as it's the antagonist. So with TMS, we were going to be measuring markers of corticospinal excitability and also neural drive from the brain through the spinal cord to the muscle. And that drive from the brain to the muscle is what we term as voluntary activation. And voluntary activation is a marker of central fatigue. So as you fatigue more, you'll see a decline in voluntary activation. So that is a key measurement for us in terms of getting a sort of a picture of what central fatigue develops in people with MS. And then the peripheral nerve stimulation, we're using 2 positions. So the brachial plexus in the sort of neck, we will use to gain maximal muscle activation. So that will be how much activity your muscle can sustain. That's the absolute maximum. And that's particularly important to express in terms of TMS. And then we will be moving to the radial nerve, so just below the elbow, just above the elbow, to measure voluntary activation for the peripheral nerve. And the reasons for that is the radial nerve directly stimulates the extensors, whereas the brachial plexus, you get the whole arm of that. So to measure central fatigue, we need evoked forces directly from the wrist extensors, and that's why we've go for the radial nerve for that. And we're also going to be measuring perception of fatigue and perception of effort, so similar to what we're doing in the scanner. And this the combination of all this data will be able to say what is happening at the brain, the corticospinal tracts, and then the neuromuscular junction in terms of fatigue in people with MS. And we'll also be able to see how perception of effort affects these physiological markers or enhances them. And for the brain, so the MRI's portion, we are going to expect to see that glutamate has risen, their baseline level of glutamate will be higher, as this is a common feature of MS. And it will probably increase further as they fatigue more, as there'll be high glutamate release. And in terms of the ASL data, we'll expect to see cerebral blood flow be elevated at rest, as people with MS, they have higher demand and their brain's working harder at rest. But then as they exercise and fatigue, we'll see a drop in cerebral blood flow. And that's common with exercise. You do see a drop in cerebral blood flow, but we'll expect to see a further decrease with people with MS. And then in terms of recovery for arterial spin labeling data, we'll expect to see an increase again, but it probably won't be as much as the controls. And then for the transcranial magnetic stimulation, we will expect to see a lower level of cortical spinal excitability at rest, and then also a higher decrease in voluntary activation. And that is exactly the same with the nerve stimulation. We will see a higher decrease in voluntary activation. And perception of fatigue. So we have trait fatigue, which is their baseline measure of fatigue, and that's going to be expected to be higher than people that are healthy. But then state fatigue, we'll expect that also to be higher in people with multiple sclerosis, and also decline further than people who are healthy controls. But this pattern of decline we'll expect to see it as the same as the controls. So it will follow the same pattern, but it will be at a high magnitude. So yeah, that's the summary of my project. So we're going to add to the literature by investigating fatigue at several different levels, from the brain to the spinal cord to the muscle, and combining that with perceptual data. So any questions?
If people at home would like to ask questions, just you can unmute. Paul, please go ahead.
Can you hear me?
Yes.
Yeah. So I'm interested in the controls. Will the controls be matched in fitness? Will they be matched in kind of cardiovascular ability? Because how fatigued you get depends dramatically on fitness.
Yeah, so the healthy controls will be largely matched in terms of gender, so we'll expect to see more females in the MS population, so we'll reflect that in our healthy control population. But in terms of the fatiguing task, it's all done relative to their ability. So the 100% maximal contraction, that's just as hard as they can. But then the second half of the exercise, that's calculated in terms of how they performed in the first block. So it's all relative to their maximum and how they produce force and what their response is to exercise. It's all relative.
But the maximum force you can produce, that just tells you how strong the muscle is, but it doesn't tell you how fit the whole body is.
Yeah, that's where the limitation comes into this project. We are just focusing on one muscle group. And in terms of the force in that first 3-minute maximum, we're including it as well to get a sense of the differences in people with MS. So we'll expect them to see probably a similar level of force to start with. Their maximum force will probably be similar, but then the decline will follow a different shape. So the decline will be steeper in people with MS, and it will go to a lower level than compared to controls.
Thank you.
Yes.
I just had a couple of comments. In fact, Paul's question cropped up in our PhD students' Viva about match people others who maybe less exercised, more fatigued, how you think about the healthy controls. So I would give it some consideration, because it could be a potential point in your advisor or a point for paganism things. So I would see if you could get a range of controls in terms of the fitness.
Yeah.
The other thing was just a comment really in terms of the language. So we've been doing a lot of work with patients who experience pain. And I know exactly why you've used the term subjective and objective here in a technical summary about an MRI project. But I would consider using induced and self-reported in patient-facing documentation or having a PPA about it, just because I think it is something that I think it makes patients feel they're not quite believed when you talk about the difference between subjective and objective things. Yeah. These are just purely comments,
No,
and I've just walked out of an hour-long PPI meeting, so it's very fresh in my brain, and I know that we want to look at these mismatches and things, but yeah, for explaining it to patients, I would talk about self-reported and induced.
Yeah, definitely. And what we're also going to do is, because the definition between fatigue and perception of effort and state levels of fatigue, they're all similar but very different. So we're going to make sure it sort of explains to participants what we mean by each measure as well.
But thank you. It's a really good introduction, because we're going to talk about pain fatigue in our as well.
Okay, that's wonderful. There's no more questions on the chat, so I think it's a good time to move on to Alex. So thank you very much, Gemma. The other slides are open, you can just open up here. And if you want, you can minimize that, the video window.
What's that, sorry?
If you want, you can minimize the video window.
This one here, you mean? Yeah, that's fine. I can keep it. Okay, so I also started my PhD in October. I was actually here a couple of years ago doing my Neuroscience MSc, which is a lot more sort of molecular, cellular in nature, like my undergrad was. But I did the fMRI module here and the consciousness module, and that made me think that maybe I want to go in a more sort of cognitive clinical direction. So I'm really happy to be here this year, working with James and Alessandro on this exciting project. As you can imagine, a lot of the time so far in this first few months has been spent dedicated with ethics, which has been an eye-opening experience, but we've got there in the end, and we're happy to receive that back on Friday. So we're very much ready to put plans into motion, and I'll try and share some of those plans with you now. So previous research has shown that low self-worth is a core cognitive symptom of MDD, major depressive disorder. And interestingly, neural markers of low self-worth are even present after symptomatic remission. So there's potential that it could also be used to predict vulnerability to relapse. And it's thought that low self-worth is underpinned by excessive self-blaming emotions, where an individual shows a bias towards emotions such as guilt and worthlessness, as opposed to indignation, which is the blame of others. So I think what we define as excessive, I should also say, so what we define as excessive could seem a little bit arbitrary, particularly if you delve into more sort of philosophical discussions about the adaptiveness and morality of self-blame. But I think the literature has typically argued that excessive self-blaming emotions, they become excessive when they are overgeneralized. So an example of overgeneralized could be to say, I'm not very good at football, therefore I'm not very good at anything. So clearly the conclusions here have extended beyond a reasonable context. So it seems that individuals with MDD are more prone to this way of thinking and to these emotions. So unfortunately, many current treatments do not account for low self-worth specifically, and treatments for depression. So, and this might result in patients not responding as well as they could do. So therefore, we think that it's beneficial to further investigate which areas of the brain are responsible for regulating self-blaming emotions, so that we can provide more nuanced targeted therapies and prognostic tools for the disorder. So in this study, we're going to use a technique called fMRI neurofeedback with a group of MDD patients to try to change their brain activity towards a sort of quote-unquote healthier pattern in a region that we've identified as being associated with self-blaming emotions. And we're hoping that this will correlate with improvements in self-worth scores and other scores from other psychological assessments. So I should probably expand a little bit on what fMRI neurofeedback is, if you haven't heard about it before. So actually, if you were here for Christina's talk a few weeks ago, she's also doing fMRI neurofeedback and using the same software as well. So little shout out to her for dragging me through all the sort of teething issues that we've had to deal with so far. But to summarize, it's a methodology that allows subjects to receive information about their own local brain activity in real time whilst they're still inside the scanner. So obviously, we present that information to them in an abstracted or simplified way, often in the form of a thermometer-like graphic that moves up and down in response to the changing bold signal or whatever it is you're looking at. So yeah, researchers will usually determine a region or a network of interest a priori, and the level of this thermometer graphic will move up when activity resembles a target pattern, and it will move down when activity deviates from that target pattern. So in this way, subjects can learn to self-modulate their own brain activity in real time with information that is obviously outside of normal conscious awareness. So this is something that cognitive talking therapies, for example, obviously don't benefit from. So it's a really exciting technique, but obviously not without its limitations. So the current study will use bold activity in the subgenual cingulate cortex, the SCC, as our neurofeedback signal. And this typically includes Broadman areas BA24, 25, and 32. But for our study, this will be a bit of a BA24, 25 hybrid that we'll be using, consistent with previous studies. I think, understandably, neurofeedback experts would be inclined to turn their noses up a little bit at us, basing a neurofeedback signal solely on one brain region, rather than, say, on a functional connectivity dynamics within a more distributed network. That is, unless there were some good reasons for doing so, and we think we have a couple of good reasons. So I think firstly, while network-based feedback signals provide a more nuanced picture of and holistic picture of whatever cognitive system you're looking at, I think they can be harder to train because the movement of the feedback signal, whether that's a thermometer graphic or something else, is contingent on multiple different regions having the right activity configuration. And secondly, we were originally planning to base our neurofeedback signal on the connectivity dynamics between the SCC and another region, the anterior temporal lobe. And whilst SCC-ATL connectivity has proven to be a good marker of self-blame in depression, results concerning which direction is therapeutic, i.e. higher connectivity or lower connectivity, they have been conflicting. And even when looking at the SCC in isolation, results are still in opposition with each other. So some studies have suggested that minimizing differences in SCC activity between guilt and indignation conditions is beneficial, whereas recent unpublished findings have suggested the opposite to be true. So I think there's a clear need at this stage in the literature and the fields to really nail down which direction of neurofeedback is going to be beneficial. And I think conceivably it's going to be easier to do this for the SCC by itself, because it's going to be able to produce a more reproducible and potent neurofeedback response. Okay, so to talk a little bit more about the study design itself. So it will consist of 3 visits. So the first and third visits, participants will undergo several clinical and psychological assessments to determine symptom changes before and after the neurofeedback session itself, which will take place at visit 2. And yeah, to clarify, all participants will be MDD patients, and we're not using any healthy controls. So the difference between the 2 intervention groups, therefore, is not based on any sort of clinical diagnosis, but instead on how we deliver the neurofeedback signal to them, which I'll explain soon. Yeah, I should have mentioned I've had a bit of feedback on this figure that it's not the most clear, and I keep meaning to change it, but I haven't got round to it. So the 4 screens the 4 sort of square screens that you can see above the top, they don't necessarily correlate to the 4 runs below them. So I think that can be a bit confusing, but just yeah, keep an eye out for that. They're just example screens. But yeah, there's also a lot of extra detail here that's not useful for you to know, but I hope you can see that it consists of 4 main runs, the scan. So runs 1 and 4 are baseline runs to assess changes in SCC bulb activity before and after the neurofeedback runs, which themselves take place in runs 2 and 3. And the guilt and indignation blocks that you can see here, the little crenulations going across the scan, they refer to time points at which the participant is encouraged to recall autobiographical memories entailing those emotions. And during those time points, the participants are really encouraged to feel the emotion during those blocks. So run 1 also acts as a sort of localizer run. So we're going to extract the 10% most activated voxels in our SCC ROI under a guilt versus indignation contrast, and then use that subset of voxels as our neurofeedback training signal. So I think the purpose of this was to identify subregions within our original ROI with greater functional relevance to guilt and indignation, given that our original ROI is quite large and the SCC is known to have quite high functional heterogeneity, and also to account for functional and anatomical variability between our different participants. So yeah, hopefully this will increase the chances of a more individualized and potent neurofeedback experience for our participants. So yeah, as I said, runs 2 and 3 are neurofeedback runs. So one will include guilt blocks only, and the other will include indignation blocks only. So at the same time as they are asked to increase the level of the thermometer, they're also instructed to recall those autobiographical memories entailing those emotions at the same time. So we're going to investigate 2 different neurofeedback paradigm designs in 2 different groups of MDD patients. So one group that we're calling intervention A will be encouraged to increase bulb activity in the SCC during the guilt blocks and decrease bulb activity in the SCC during the indignation blocks. And the other group, intervention B, will be encouraged to do the opposite. So decrease bulb activity in the SCC during the guilt blocks and increase SCC bulb activity during the indignation blocks. So importantly, if you remember, I said in the first run that it's a localizer, which is under a guilt versus indignation contrast. So this means that intervention A group participants are actually being encouraged to enhance differences in bulb activity between guilt and indignation, whereas intervention B participants are encouraged to minimize condition differences between guilt and indignation. Yeah, I hope I haven't lost you there, because that's a little bit confusing, so feel free to jump in at the end if there are any questions there. But I think it's important to reiterate that both of these training directions while they are different to each other, even opposite, they do reflect activation patterns that have been observed in healthy control populations in previous papers. So we're confident that that neither intervention will incur any significant worsening of symptoms, particularly as we're only doing a single neurofeedback session with each participant. So instead, we hypothesize that both groups will be able to self-modulate their bold activity in the SEC towards a target level, and in at least one of the intervention groups, this will correlate in some way with increased self-worth scores. And yeah, importantly for this study, we're not providing participants beforehand with any psychological strategies that might help them to increase their thermometer level as they're in the scanner. So instead, we're opting for a more self-guided approach, and hopefully by trial and error, participants will learn which ways of thinking will help them to increase the level of the thermometer. Yeah, next step. So as I said at the start, a lot of time so far has been spent wrestling with ethics, and yeah, we've got there in the end. So yeah, whilst we've been waiting to receive ethics approval back, we've had a bit of breathing space to do some other sort of preparatory work. So yeah, a lot of my time right now has been spent reading and writing. I've been told it's a good idea to get the ball rolling, writing introductions and lit reviews and things like that right now. I'm making a Qualtrics pre-screening questionnaire for prospective participants, liaising with the software developers to get some extra functionalities added to the software. Yeah, writing up some more detailed documentation for software and all the other things that all of you are, I'm sure, more familiar than I am with setting up a new study. But yeah, I'm also conducting a little separate analysis at the moment, independent component analysis in ICA, which is a little bit on pause at the moment, but we're still hoping to do that. And we're going to harness an ICA on a previously published fMRI dataset of self-blame in order to determine more clearly the functional architecture of self-blame and other blame in a model-free manner. And originally it was hoped that this first analysis could then in turn inform our signal selection for the subsequent neurofeedback, but unfortunately for a number of reasons that wasn't going to be feasible. But yeah, I'm looking forward to all the rest of the fun to come. Yeah, thank you for listening.
Does anybody have any questions? Yes.
I mean, I'm sure this is being covered by the ethics, but just also the point, you're using patients who are already self-critical and self-blaming, right, and showing them feedback and of their ability to manipulate their brain. Perhaps they could blame themselves for not being able to do the task.
Sure.
What are you going to do about inducing psychological distress? I'm sure you must have considered this.
Yeah, of course. No, it's a great question. It's obviously something that we've been wrestling with for a long time, the sort of potential negative consequences of this. I think Roland, who I didn't mention at the start, he's been doing a lot of this work at King's already, and he's been able to show quite clearly that there are no negative consequences of this in people. We're also we've got quite a nuanced exclusion criteria to exclude some sort of subtypes of depression who might be more might have a greater proclivity to react negatively to the neurofeedback, whether that's people with sort of history of violence, that they're more likely to experience negative reactions to the indignation training or whatever that is. And we've also obviously got the safeguarding steps in place should there be for whatever reason something that goes wrong. But no, it's a good point.
Does anybody on the Zoom have a question? Paul?
Yeah, thank you. So there's several factors that might influence how big an effect you get. One is that sometimes when people go in the scanner, they're terrified, and so they're not acting as they normally do. And secondly, some of them may engage more than others with the whole process of doing the neurofeedback. So I wonder if you identify those people in your pre-psychological assessment, you could then perhaps focus on the ones who are more likely to respond positively to your intervention, and you may get a bigger effect size that way.
Yeah, sure. No, it's another great point. I think we will be trying to screen for claustrophobia and anxiety in potential participants as well, to deal with that first point. In terms of your second point, the effort that people are likely to put in, I don't think we currently have anything in place to screen for that at the moment. I'm not sure what would be the best way to go about that. But
you might just be able to ask them, what do you think about this idea?
sure.
Is it interesting or boring?
Sure, that's a simple way, yeah. But no, it's a good point. It's definitely something that could be a confounding factor. So yeah, thank you.
Just a brief question. So you have is that okay, Chris?
Yeah, go ahead.
Hi, Alex. So you have two conditions, is that right? And do you have any thought which condition works better as a therapeutic?
Yeah. Yeah, thanks for the question.
Yeah.
That's the big thing. So we originally were calling one group the treatment group and one group the control group, but actually after we were wrestling with it a little bit, it was quite clear that from the literature it wasn't obvious which direction was going to be the more therapeutic. So that's why we've renamed the groups, reconceptualized it to intervention A and intervention B, and we're interested to see which is going to be the most therapeutic. So we're not sure. As I said a few slides ago, the literature is a little bit in conflict as to which is most therapeutic. So that's what we're really trying to nail down.
So I have a question more technically about your localizer. So you've got these two conditions, and you're going to be localizing a region.
Yeah.
Has that localizer been used before, and does it reliably produce? I guess what you want to think about is what happens if you run that, because you're going to be running that at the start of your scan.
Yeah.
And then you want that to define a region which is necessary for the subsequent steps.
Mm-hmm.
What do you do if that region is not present? Is there a way that you can intervene, change thresholds, make it smaller or larger? And another point is, if you end up with a very, very small region, and you've got feedback going on, and the participant kind of gradually moves their head ever so slightly, even by a few millimeters, because the region is so small, that is going to look like a reduction in activity. And
sure.
The participant could learn, even unconsciously, that slightly moving your head by a couple of millimeters produces the feedback on the screen. And I guess I wonder if other people have used this method before with a very small region.
Yeah, sure. I mean, I think I'm correct in saying that as we're selecting the 10% most activated voxels under that contrast, that the ROI will be of a similar size, not too small or too large than the other ones. And yes, Roland's group has previously used that same localizing strategy, selecting either the 10% or 50% most activated voxels in that ROI, and he's shown that to work quite nicely. But yeah, we don't have any plans in place to sort of alter that ad hoc if needed. But I don't think that we'll be in a position to need to do that. But yeah.
Are there any other questions before we move on to Jess's talk?
Just one more quick question. Just a practical level. How this can be used as a treatment, because I treat patients with epilepsy with biofeedback. The patient have to go through 12 sessions over the course of 1 month, and how one treatment, let's say, supposing this is effective in 1 year, and in a practical level, do you think that one or let's say how many sessions like this could be necessary to treat patients?
Yeah,
we don't know yet, and but this is something it's good to think ahead, I think.
Yeah, so I think that a single neurofeedback session, as I said, is a little bit hopeful to to incur any sort of long-term changes. And I think that's why for this study, our primary outcome measure is going to be bold activity changes, and our secondary measure will be the psychological and clinical changes that would be great if they happen. We're not entirely convinced that we will find much of a change, if anything, because we are looking at one neurofeedback session only. So yeah, this isn't so much of a clinical feasibility study as a sort of initial feasibility. So yeah, I think for sure multiple neurofeedback sessions looking ahead would be required to incur long-term.
Yeah. Thank you.
Thank you very much, Alex. Okay, so let's move on to Jess's project.
Okay, so do I just
If you just alt tab, it should be up. It's already open in another.
so alt tab?
Yeah.
Oh,
If you.
yeah, there we go. Okay, sorry.
Or you can just minimize it and
Yeah.
they're all.
Yeah. Oh, I've done something terrible now. There we go.
There you go.
From the beginning. Slideshow. Okay. Hello, everyone. My name is Jessica Eccles. I'm a clinical academic liaison psychiatrist in the in neurosciences at BSMS. I want to talk to you about a pilot scanning project that we are hoping to do in conjunction with Alessandro, Itamar, and Eccles, about the efficacy and mechanism of a novel treatment for a condition called fibromyalgia. And for those of you who've been in the group a while, you'll remember that our postdoc Christy and our PhD student Marisa were involved in a big imaging study of pain and fatigue in fibromyalgia and ME/CFS that ended just before lockdown. So the efficacy and mechanisms, so the whole purpose of this work, this pilot work that we want to get some feedback on really, is to boost our chances of being successful at an NIHR, so National Institute of Health Research grant called Efficacy and Mechanisms. And it's an unlimited budget, but we want to show good value for money and that we are on the cutting edge or leading edge of science. So, yes, and so this proposal is being led by myself and Harm, the professor in general practice. For those of you who are not sure, what is fibromyalgia? Fibromyalgia is a common pain condition affecting probably at least 5% of the population. It involves widespread pain and fatigue and is often quite debilitating, and it's poorly understood by doctors, it's poorly understood by the media, it's poorly understood by patients, and it's likely that there are a lot of different processes going on. But it was traditionally thought of, and some of the main hypotheses about fibromyalgia are basically that it is a functional disorder, so a sort of psychological, physical representation of psychological distress. I'm not convinced by that interpretation, and I think it's multifactorial. And I'm just going to show you some interesting behavioural work that we did in our previous study that sort of implicates some things that might be going on in fibromyalgia and this related condition, ME/CFS. So thank you very much to Lisa for producing these plots. But essentially, we induced pain and fatigue in patients, but not healthy controls, by an autonomic challenge, so a challenge to the involuntary nervous system. So that's the bottom line. So if you have fibromyalgia or ME/CFS, you're more likely to get challenge-induced fatigue. And that relationship is in fact mediated by higher levels of inflammation in the patients, which predict fatigue level. So we have here a sense of autonomic mechanisms potentially in symptoms in fibromyalgia or ME/CFS, and a possible inflammatory component. We also have, coming to pain, and it's very hard to tease apart what the primary outcome measure should be. We have fibromyalgia patients and ME/CFS patients compared to controls have autonomically induced pain, and that is mediated by the number of connective tissue features associated with joint hypermobility. So there are some bodily constitutional factors that are going on that may be related to the expression of pain and fatigue in these complex groups. And then this is some initial work from Marisa, our PhD student, who looked at transcriptomics between patients and controls, and found that between the groups, and again, the question about how matched the controls is a good one, it found that there are different genes upregulated and downregulated. So a large proportion of genes upregulated in patients compared to controls. This is to do with gene expression, and some downregulated genes. And when we look a little bit into the common pathways, is Alessandra in the audience? Alas, not. Never mind. Mitochondrial metabolic processes come up as a possible, that should say patients versus controls, sorry, not controls versus patients, as a possible difference between the control group and the group with fibromyalgia or ME/CFS. So why do we want to do a clinical trial of a new treatment for fibromyalgia? Well, as I said, this is very poorly understood and is often thought of as a bit of a heart sink condition, in that there doesn't seem to be a treatment. But the more you understand about mechanisms, the more likely you are to have targeted treatments. And it may be that there are targeted treatments out there already that we could repurpose cheaply without having to undergo drug discovery trials. And treatment is really important because, and Lisa and I were just running a PPI group about this, because these patients really do feel dismissed and forgotten about and left without any sort of intervention and often hope. So is it enough to know that a treatment works? And this is a sort of a philosophical question. Or do we actually need to know why? And can we subject our participants to quite complicated, uncomfortable processes to find out why? And our PPI group tell us that yes, finding out why a treatment works is important, not just for kind of natural curiosity, but also for how these complex conditions are understood in the wider community. So I think we're on the right track to try and do some work in this space. And I don't know if people in the audience have heard of naltrexone. I mean, the psychiatrists amongst us will have heard of it, but in the context of substance misuse. And naltrexone is an opioid antagonist at normal doses, and is used in the treatment of drug addiction. But and the normal dose, I think, is 40 or 50 milligrams. But there's a lot of traction about the possibility of using lower doses of naltrexone for conditions like fibromyalgia. And in fact, not just fibromyalgia, things like long COVID, ME/CFS, postural tachycardia syndrome, all of the sort of complex conditions that I am and others are interested in here. So what we propose is to do a clinical trial of low-dose naltrexone, a crossover trial. So let us say we have 100 participants, which is what our current power calculation tells us, and they will have low-dose naltrexone for 12 weeks, and then a washout period, and then placebo for 12 weeks, but obviously randomized and crossed over. So each patient will act as their own control, and I think ethically that is the right thing to do. But in order to determine the mechanisms of how low-dose naltrexone will work, each participant, so there are no healthy controls in this, the patients are their own controls. The participants will have 4 lots of testing. So pre-post low-dose naltrexone, pre-post placebo, and we will be looking for a variety of factors that could lead to the success of low-dose naltrexone. The initial trials suggest that it is promising, but they don't tell us anything about the mechanism. Okay, so Alessandro kindly introduced us to this study when we were thinking about what would we like to image in the brain to see how low-dose naltrexone might work. So low-dose naltrexone is thought to have anti-inflammatory properties. So it's been used for multiple sclerosis, Crohn's, various things. But what this rat study suggests is that in a dose-dependent manner, naltrexone modulated expression of this mTOR, which underlies the cell metabolic phenotype, regulating microglia immune properties and adaptation. By modulating the phenotypic features by metabolic switching of activated microglia, naltrexone was found to be an effective and powerful tool for immunometabolic reprogramming and could be a promising novel treatment for various neuroinflammatory conditions. So that is a bit of a mouthful, but we know that the microglia in the brain are important in psychoneuroimmunology, inflammation, and what this suggests is that naltrexone, low-dose naltrexone, is working on the metabolomics, so to speak, or not the metabolomics, it's something else, on how microglia metabolism works. So what we were thinking is it would be very interesting to capitalize on the skills in the imaging center to actually do some phosphorus imaging, which is, so for those of you who are not biochemists and to learn the Krebs cycle and all of that at medical school, ATP phosphate, we want to look at how phosphorus is metabolized in brain. So we'd like to do some phosphorus imaging. We would also, and this relates to what Gemma was talking about, you know, I talked about those autonomic induced pain and fatigue challenges that were related to inflammation and hypermobility. We suspect that there may be influences on cerebral perfusion as a consequence of inflammation and maybe underlying constitutional features. So we would like to do some ASL to see whether low-dose naltrexone has an effect through metabolism, perfusion, and inflammation. So this is all very, very pilot. We're going to do 12 scans, basically to optimize the sequences, have a combination of patients and healthy controls, really try and put in action the phosphorus imaging sequences alongside these. We're thinking, is it feasible to run these sequences in a patient group? What issues are there? So Lisa has been leading on some work that shows that patients with chronic pain and fatigue are much more likely than those in a comparison group to have neurodivergent features such as potentially ADHD traits. Does movement, is that an issue? And what sort of challenge should we do in the scanner so that we can dynamically look at these imaging markers? Should we do finger tapping? Should we do visual stimulation? There's lots of questions. We're actually questioning whether we're going to look at fibromyalgia at all, or whether we're going to also look at MECFS. Yes, there's lots of food for thought, but we hope that, and this is just one part of other mechanisms that we'll be doing alongside transcriptomics and possibly skin biopsies. So we want to get it right. If we were successful, we're looking at probably doing 400 hours of scanning over a 2 to 3 year period, which would be quite a big thing. So any questions or comments? I don't know if Itamar, Iris, or Alessandro is not in the audience. Are Itamar or Iris? No. Nick or Samira?
Yeah.
Oh, great, because they will understand much more about the phosphorus imaging than I do.
There is a couple of questions. There's one from Guy and then Mara. So Guy, do you want to go ahead and ask your question?
Thank you. What dose will you be administering to the patients? Would it be 4.5 milligrams? And also, would you have a ramp up phase to that? Because I know that the LDN Research Trust, I think they advise it to do it in a ramp up phase to build up to that 4.5 milligram dose.
Hi, Guy. I don't think we've met.
Camera's up there if you want.
Oh, camera's there. Okay, that's an important thing. Okay, so this is something we have put to our, so low dose, we are thinking 4.5 milligrams, but you're absolutely right. In the literature, some people think low dose 1 milligram, some people think 9 milligrams. It's all, the literature review that we did, basically systematic review, suggests 4.5 as the optimum dose. We have decided against a ramping up in conjunction with our PPI group, who are patients, majority of patients who have actually taken low dose naltrexone, or been prescribed it, but not in a, in a off license way. So that is the plan, but we may have, we're open to all sorts of suggestions and ideas, but that is, when we put it to the patients, they said they thought it was best to just go at one dose.
Okay, thank you. And will you be looking at the dopaminergic systems and endorphins, things of that nature? Because I know that's related to might be related to the mechanisms of action as well.
So we have an expert patient in our grant application group, Monica Bolton, who published with harm a case study of her own experiences in BMJ Open of taking low dose naltrexone for MECFS, and she has been talking to me a lot about endorphins, and we have to work out how we measure them and what we would be looking at. We're currently everything's up for grabs, and we would be really interested in any suggestions or methods that people think that we might be able to use locally, or collaborations. So Guy, sorry, what's Guy's surname?
Mitchum.
Ah, Guy, which department do you work in?
I'm in the School of Psychology, but I follow you on Twitter. I tweeted you a few times. I'll message you on there.
Ah, thank you. Well,
Yeah, sure.
that's fabulous.
Okay, brilliant. Thank you.
but we're definitely thinking about endorphins, and also the practicalities of doing this study. So there is a lot of work, well, not a lot, but there's a bit that suggests that low dose naltrexone is probably effective, but no one knows why. Any other questions?
Yes, Mara has a question.
Oh, yes.
Go ahead, Mara.
Hi, Jess. Very exciting. I just wanted to, maybe you don't know the answer to this, but it's about the phosphorus. Maybe you've said it and I missed it.
I didn't say much.
So you're planning to measure it in the brain? Is that what the plan is?
Yes, brain. Yes, because Iris was originally like, are you measuring it in the muscles? And no, we are planning on measuring it in brain.
So, I mean, just thinking about the equipment that is available in CISC, the phosphorus coil that we have is actually a surface coil, which means that you will have to sort of focus on one area of the brain. Do you have any ideas of where, and are you planning to measure phosphorus changes during a task or just at rest?
So I can answer the second question better than the first. So my understanding from discussions with Itamar, Iris, Nick and Samira is that we will be doing a challenge like finger tapping or visual stimulation and comparing that dynamically to baseline to try and work out what's going on. So that's the second part. The first part, what part of the brain are we going to localize on?
Well, if I may say, given your answer to the other part, probably, if it was me, probably I would, if it makes sense from the point of view of your hypothesis, maybe if you're using a visual stimulus, probably visual cortex is going to be an easy area, well, easier in the sense that with the surface coil, you should have enough sensitivity to look there.
Yes. I actually, and correct me, Nick and Samir, if I'm wrong, but I think this is the point of the pilot, really, is for us to see, because I don't think anyone has yet done much phosphorus imaging in brain here, have they? I might be wrong, but
no. Part of the reason is because the coil is a surface coil and not a head coil, but also I think, at the moment I think we only have spectroscopic sequences to look at the phosphorus.
Are Nick and Samira able to jump in at this point?
But it was
yes. So we've got we've got spectroscopic sequences, and I think you're right. The pilot, we did flag up this issue that it being a surface coil, you couldn't just sort of have the freedom of doing the whole brain and placing the coil in a single position. So having a hypothesis-driven placement of that voxel would be important. But like Mara says, looking at the back of the head is ideal, really, because they just lie their head on the phosphorus coil when we do the scan.
Nick, is it a localized sequence or is it just I can't remember, because we did it in muscle. Yes, it might be a localized sequence.
Yeah.
Okay. All right, thank you so much. I've got to run, but it was really good to hear all this.
That's really useful, but I think if we're thinking that it's the metabolism of microglia, obviously microglia are pretty much everywhere in the brain, picking that particular area where we're doing the challenge, hopefully that will show us what we want.
Thanks.
Thank you. Are there any other questions, Chris?
As far as I can see, is there anyone else got any questions before we wrap up? Nope, doesn't look like it. So thank you very much, everyone, for a fascinating series of talks. Thank you, everybody, for coming.
They're all sort of interrelated in some way. Yeah. Okay, thank you.